Setting up a new experiment on Rosalind is incredibly easy. To begin, I'm going to start on my project tab. So I'm going to go ahead and go into my internal demos project, and I'm going to click add a new experiment. Once I do, I can see three different types of experiments: a partner recommended, which would be one where it's sponsored like NanoString or Lexogen, or I could pick to design a new experiment, which is what I'm going to do here now. And this is for RNA-seq and single cell and ATAC, ChIP-seq, lots of different things we can do. So I'll go ahead and click design a new experiment. Give this experiment a name. So with that, we're now going to set up a new experiment. I'm going to begin by choosing the method. So this is sequencing. I'm going to do this with a process counts RNA-seq for the purposes of brevity. I'll go ahead and click next. Immediately after I do that, Rosalind says, great. Well, I'd like your counts file. So I've got one on another screen. I'm just going to drag it over here. I've got my counts data file. Drop it on. Rosalind will quickly look at that file and make sure that it's looking okay. We see 32 samples, 25,000 gene rows, and I have an opportunity to add some metadata. Now, this follows the NCBI biosample bioproject data model, which is great to have a consistent ontology, and it's great to even simplify data submission when it comes time, should I choose to do that. So I'm going to grab my attribute file, which I have over here. This is a very simple file, and I'll also guide you through some of that step here in a moment. Rosalind immediately found three attributes that nicely matched everything that were here. If I had something off, so for example, I was missing a sample or the data didn't connect, I'd get a few error messages. Rosalind would tell me, hey, you're missing a sample, or this data doesn't look right. So I received very quick validation. And as an example, let's say that I said, well, I'm not sure if it's normalized or raw, but maybe it's normalized. I don't remember what the bioinformatics team told me when they gave me this information. Well, Rosalind's going to look at that data and immediately flag and say, the data you have here really looks like it's raw. Are you sure that's not what you have? And gives a quick opportunity to understand that. Obviously, that is what I have, so I'm going to go ahead and flip it back over just to say we try really hard to make sure there's sufficient intelligence to help guide you through this process to make sure you get quality results and avoid some of the typical pitfalls that we've heard from scientists and biologists over the last several years. So with that, I'll go ahead and click next. At this point, Rosalind's going to want to know, well, what's the species? Of course, we have many, many, many species in Rosalind. This happens to be human. And I did bring this in using the UCSC HD19 symbols and genome build. So I'm going to go ahead and click next. Rosalind's going to validate that my data file and all the gene symbols that I had used do conform to what's expected here. So already I'd added the attribute file in the beginning. So if I hadn't, I would be able to get this built out now. So we can see that I have cell line was added from the attribute file, treatment, and time course. If I wanted to, I could go ahead and add more. So for example, let's see, I could add age. And notice we get a different color because we're adding this now. So if I want to do a custom one, like let's say I just wanted to call this custom, notice we have custom field 1, 2, and 3. I wanted to do custom field 33, which doesn't exist. If I just hit return right now, Rosalind will add and create custom field 33, which becomes a custom attribute that I can use inside of my experiments. Now by clicking on it, it makes it go away. And so that's now gone from that list. But again, as I mentioned, I can just put that right back in, custom field 33, and there it is. So already in my user database that's accessible for me in setting up my experiments. I've already configured this with these three values, so I'm not going to do that. I'm going to just go ahead and knock those off, keep these three primary ones. And if I wanted to, I could ask Rosalind to generate an attribute file for me. And so this would take all the sample names and the information that I currently know and would export that into a CSV file. And then if I wanted to take where I had these three attributes I've already added, or I could add more, those would all be column headers. And then I could copy in and paste in all the associated sample annotation associated with each of these column headers. So the time course values, the treatment values, the cell line values for every sample. And then that would be available to me when I re-upload my attribute file. This is already set, so I can go ahead and click next. I can now see all of the values that were included in that attribute file. So, and I have the ability to delete any individual sample that I might want to here. And I can see all the attribute values or the values within the first 100 rows of the counts file to confirm everything is as it should be. If I need to change this, I can upload new files or edit my attributes. This is all fine. I'm going to go ahead and click save. And now we're ready to create a new comparison. And as I've said many times, it's always best to don't use sample names to build comparisons. It's the best way to make mistakes. It's better to describe the biology of the experiment, use the NCBI biosample data model that we enable for you in Rosalind, and make it easy. ARS 1620 versus DMSO, treatment versus control. Control is always on the bottom. If you need to modify, you can click edit and see all the samples that are associated in that grouping. If you want to, you can remove individual samples there, or you can also compound these values. So I can come and say, well, I want H358 only on both lines. This reduces down as we do this. And I could add a covariate variable. That's been covered in another video, so I won't dwell on that now. I have the values I want. I click save. As soon as I do, Rosalind comes back. I can create any number of comparisons. Once I've done, I click next. And with that, Rosalind will launch and begin processing. And in a short 20 or so minutes, the analysis will be completed without having to write a line of code, know anything about bioinformatics, just getting my data files in and immediately diving in to my experiment.